This project has been a lot more challenging than we anticipated, and it's taken 3 years longer than I confidently predicted 3 years and 3 months ago. So I'm very pleased to be able to at least present some of our preliminary results on behalf of the consortium that did this work. So I think in this audience, I don't have to belabor the point of the huge amount of genomic diversity in Africa and the contradictory lack of information in the key databases with genomic information. But I want to stress that it's particularly the case with respect to structural variation. And this is a problem because not knowing what structural variants such as deletions, copy number variations, insertions, and their whole range of others, not knowing what those variants are has important consequences because of the biological importance that structural variation has. And when we lack information about structural variations in our target population, it has a number of problems downstream in terms of various bioinformatics analyses. So our goal was, we thought, the modest one of building a catalog of high-quality structural variation calls. High quality in the sense that we wanted to make sure that there really were true positives rather than trying to maximize the number of positives, and also to explore regional variation. And we had this opportunity because of the novel data that was generated by various H3A projects and initiatives. And this gave us a number of groups the opportunity to get together to work on a common project to explore this variation. So we had about 1,100 samples in total. About half of those were novel, had not been studied before for structural variation. And on the map, I've got showing you where the novel data came from, which countries they came from. And on top of this is the public data sets, mainly 1,000 genomes data. So you can see we were able to capture a lot of the countries in Africa, obviously some major gaps. And if we do a PCA of the single nucleotide variation in the data, you see the same picture. We capture a wide variation of genetic diversity in Africa, but with some significant gaps. So we could use this 1,100 samples to try to build this catalog, and it's proven to be challenging. Structural variation has had a lot of attention over the last few years. A number of groups have built some excellent tools with different approaches for detecting structural variation. Some really sophisticated algorithms are out there, very computationally intensive. But the truth is that, at least compared to single nucleotide variation, these algorithms all perform poorly with respect to accuracy. So one approach that's been common in the field is to do ensemble calling, where instead of applying one tool to do calling, you apply many tools to do calling. This, of course, comes at a significant computational cost. We probably use close to 1 million CPU hours so far to do the work that we have. So in total, we used 7 tools. The main part of our workflow, I won't go through it all, but the main part of the workflow, which was to look at structural variants that are less than 1,000 base pairs in length. We used 5 tools, and then we used a tool called Survivor to combine the results. And essentially, we said, for a variant to be accepted, it must have been called by at least 3 of these 5 tools. So we did this, and we had to do several iterations, and eventually we came up with results. Now, there were some methodological challenges we had, and you're fortunate that I've got a short period of time and a very stern-looking moderator. So I'm not going to drone on as much as I'd like to about Wynn looks happy about that, but I'll corner him at tea time and I'll bore him. But I want to give you some flavor of the problem. So let's take an example of a single nucleotide variation. We've got a reference genome, and we've got 2 calls. Let's assume these calls are from different individuals. They could also be calls from different callers. So on the left-hand side, we've got a T in the reference genome. For individual 1, we have a G and an A for individual 2. So immediately you can tell those two calls are different to the reference genome and they're different from each other. And on the right-hand side, we have an A on the reference genome, and call 1 and call 2 are both C. Immediately we can say they're different to the reference genome, but they're the same call. There's no ambiguity. The left and the right, there's no ambiguity. The same does not apply to structural variation. Let's suppose call 1, we find a structural variation that's 7,000 base pairs long. And call 2, we find a variant that is 7,002 base pairs long, and maybe there are 1 or 2 minor differences in the middle. Is this the same call or not? And you can see that depending on the differences at one end and the other end, it will be very clear that they are or not the same call. But it turns out in practice, there's a lot of gray area. So determining whether two calls are the same is a challenging thing to do, both, I think, biologically and computationally. So these are our results. We found about 92,000 structural variants in total, less than 10,000 base pairs in length, and a few hundred that are longer than that. About 15% of these are novel on a strict definition of novelty, but as I've indicated, determining what's novel isn't so easy. So these are structural variant regions rather than structural variants. And the data is available, both the raw calls and the consensus calls are available, and we hope to have a searchable database available as well, so it's a resource that can be used by researchers. So you can find population structure in this data. We expected this to be the case, so there is considerable variation across different regions of Africa, and it's important to take that into account when you're doing projects in different regions, that your background allele frequencies and what's out there is going to be different. The different tools performed very differently. So if you look at the key 5 that we used, you can see there's almost an order of magnitude difference in the variants that were called, and with varying degrees of accuracy as well. So we did quite a lot of work trying to understand what combinations worked and which didn't, and that's how we came up with deciding that 3 out of 5 callers would be the right number to use. So just to describe some of the things we found, all the individuals had several thousand structural variants in them. Fairly narrow region from about 2,800 to 3,400. The lengths of the structural variants, not as interesting as we hoped, because we realized after the initial analysis is that this does not reflect biological reality, but rather the peculiarities of the variant callers that we used. So for example, if you look at the insertions, you'll see there's the strange bimodal distribution of the lengths of the insertions that we found. That is not a reflection of the true lengths of variants that are found in individuals. It's a reflection of the fact that first of all, calling insertions is difficult. Some of the callers are good at calling longer variants, and some are good at calling shorter variants. So when you do the ensemble calling and saying at least 3 callers must call it, you find that at least one falls out of the window every time you do this, and that's why you get the strange length distribution. So there are some other anomalies in these results as well, and it's important to bear that in mind. The structural variations are found throughout the genome. We were surprised by the number of variants that at least overlap partially into a broader gene region. This may reflect the fact that once you're dealing with long structural variants, then it becomes less unlikely that a variant is within a gene region. But it is higher than what we would have expected. Also, as expected, if you use some of the standard tools like NOT-SV or VEP that are used for annotating structural variants, most of these variant regions are benign, but there are some pathogenic or likely pathogenic variants that were found as well. And this is probably an area where more work needs to be done to make sure that we are taking into account African data. This may not be reflective of what's out there. But I think we're hoping that the availability of data like ours and being able to build up information about African genomes is going to help this type of analysis in further. So if you're interested, we've done in our paper, we've got an analysis of the pathogenic regions and what they do. So some problems, very different results from the different tools, much more than we expected them to be. And I think this does show that methodologically there does need to be improvement in this area. Computationally, it's extremely intensive, and there are I think, methods of trying to be able to improve performance. And if you thought VCF was easy, when you start looking at VCFs for structural variation, you'll see it's a really complicated area to get into. So our next steps, we're hoping for new data. There's a project called Agenda that is sequencing 1,000 new African samples from underrepresented groups. I'll be knocking on Nikki's door for access to PAGRA data when it's ready. And if anyone else has got data, we're very nice people, so please share your data with us. Obviously, we want long-read data. We've got a small amount of long-read data that we want to use for benchmarking, but ideally, we need to bring a large amount of benchmark of long-read data for accurate calling. So there are a lot of people who've contributed to this project, and I particularly want to thank my co-lead on this, Sine Lombard, and the data providers and our funders.
Thanks so much, Scott. Is there any questions?
Thank you, Scott. Great job from you, Macquarie University. I just want to hear your comment about the high percentage of those structural variants found in intronic region.
Yes, so maybe intronic is the wrong word to use. It's in from say 1,000 base pairs up from the 5' to 1,000 base pairs down from the 3' region. So perhaps it's the wrong label to have used. It is a bit surprising. We have double checked it. I think it becomes less unexpected when you consider that if a structural variant takes up is relatively long, then it's not so unlikely that it's going to overlap with a gene region, as opposed to SNP, which is just a particular point. So it becomes less unlikely as a result. But it's still higher than we would have expected.
Thank you, Scott. So I have a question. I have seen in one of your plots that you have short deletions, a few short deletions and maybe more insertion. Sure.
There we go. Yeah.
You see the number of deletions are very short and
yes.
Insertion. How do you explain this? And are the insertions the same?
Yeah, so that's a really good question, Alia, and I'm not sure what is a biological reality and what the tools are capable of doing. So with the insertions, I think that's a really, you can't interpret those insertions as biological reality. That's just a reflection of what the tools can provide. So the same thing is true with deletions. Are the methods capable of detecting long-range deletions? And we could see, I mean, one of the reasons that we cut 10,000 as the upper limit is we started to see a number of deletions that were significantly longer than that. And on analysis, they looked a bit dubious to us. So I can't explain it other than I think it's the tools are still need elaboration, and we need access to large scale long read data to have more accurate analysis. So if you can do direct, take the reads and then from the reads themselves, rather than through the other approaches, find variants, you'll be able to get more accurate idea of the length of the regions.
And that be because of the reference.
Oh, that could be as well. So we did build 38. And so part of what we'll do with the agenda project will be to do at least some preliminary analysis. Does it make a difference whether we use T2T or build 38? And we may do some pan-genome work. I mean, we'd love to do the pan-genome work. It just depends on our bandwidth to be able to do that.
So we're going to take 2 more questions because Scott, you seem to be very popular and we're going to have to get everyone to chat to you during lunch. So just 2 more questions.
Yes, we love you, Scott. And I concur with your call that we should all do more long reads and avail more long reads data, because for this type of studies it's absolutely critical. I like this talk, and I wonder whether you have used exactly this kind of statistics to maybe try and relax your constraints, your 3 out of 5 constraints. Have you tried to see what this would look like if you say just like, if any color calls something, let's consider it, and how would you affect the distribution, specifically the binomial distribution of insertions?
Yes, so I haven't done the any one. I have done the individual, looked at this if we did that for the individual callers themselves. And then you don't see the anomalous results you get here, but you see a more expected distribution, a more natural distribution, but the distributions are all general, are all different from each other because they reflect that the different tools use different approaches and therefore are capable of detecting different types of variants. And so that's a strength of doing ensemble calling, but the weakness of it is you can't then claim statistical representativeness of the biological reality as a result of that.
Okay, last question.
I have a very simple question. So I'm assuming these are healthy individuals?
So these are from various projects. So many of them are population cross-sectional. Some of them had some conditions. So perhaps the biggest single one is there are people who have HIV.
Right.
But I would say that in general, these are people who are, for our purposes anyway, are population controls. They're all older individuals. So it's unlikely that they have any really serious genetic disorder. Otherwise, they wouldn't have been captured.
Right. So now my question. So you think you would probably draw in a bathtub? So if you brought in mutation-heavy population, cancer population, do you think this disparity would exist?
Oh yes, I think that probably would change. Although don't forget, even in people who are ill because of a structural variant, it's only one structural variant in that individual that's going to be the problem. It's not going to be 300 structural variants in that individual. So I'm not sure it would statistically change if you brought in a lot of people with different genetic disorders. But it might change somewhat. Part of the purpose of what we're doing, one of the projects is studying the impact of structural variants on developmental disorders. So part of what they wanted is a catalog that they can use to do filtering on. So when they find in a child who's got a structural variant, they want to know, is this variant something that is seen in African populations, or is it something that could potentially be novel in this individual and therefore implicated in the disease?
Okay, so Scott, you can be very popular at lunch. But thank you very much. Have a good day.